My name is Alain Kohl and I am Professor of Arbovirology at the MRC, University of Glasgow Centre for Virus Research. The Centre for Virus Research, as it is today, is born out of what used to be the Institute of Virology, which was in the main university campus in the West End. And we're now here in a new building, and this is a structure that is much bigger. We're over 200 people here. We have a number of labs that work on different topics in virology that goes from structural virology to work on SARS-CoV-2, to innate immunity, to the arbovirology with vectors like mosquitoes and ticks that I'm involved in. So we have a very broad range of expertise and people here at CVR. So my lab works on arboviruses. These are viruses that are transmitted by mosquitoes, ticks, midges to humans and animals. And we are particularly interested in understanding how transmission works. And so we develop a number of tools to manipulate viruses to understand these processes better.
Hi, I'm Akira Alexander. I am a researcher here at the MRC Centre for Virus Research at the University of Glasgow. I am investigating a virus called Toscana virus, which comes from the countries surrounding the Mediterranean and is spread by the bite of female sandflies. In my work here, I use quite a lot of Promega tools, in particular the luciferase-based assays, cell titer glow, vanilla and firefly luciferase, nanoluciferase, and now HiBiT. There's a protein in our virus which has an unknown function. So I tagged it with HiBiT to see when and where it was expressed during the infection. I have tagged HiBiT on the N, C and middle of the protein I'm interested in and generated a virus which has the HiBiT sequence. I have then infected mammalian and insect cells with the virus, and we then lyse the cells at various time points and measure the luciferase output. The advantages of using HiBiT are quite simply the size. It's very small, the viral genome is very small, so the tag that I use has to be small. And because HiBiT is luciferase based, it is real time. I do use other tags as well as HiBiT. I use the V5 tag and the MIC tag as well, but because they are antibody based tags, I cannot detect them in real time. That is why HiBiT was a benefit.
The advantage of using a shorter sequence like HiBiT is that if we want to produce a virus for reporter gene experiments, we don't need to dramatically increase the length of the viral genome, and we don't need to replace a viral protein by a luciferase, for example, because that does have deleterious effects. To some extent, there could be attenuation. Virus replication might be impeded in one way or another. And so by using a very short stretch that we insert, we hope that we avoid largely these deleterious effects on viruses. Being able to then probe the tag also with antibodies, for example, makes it possible for us to then also look at expression in different manners, to look at interactions and so on. So we probably get better data really by using a shorter tag that we can also use as a reporter. I would always sort of point out that this specific technology has been extremely useful to our work and saved us a lot of time.
So here in the lab we use the GlowMax plate reader, and I quite like it because it comes with all the settings pre-installed for all of the Promega kits, and it's quick and easy to use.
I've been using Promega luminometers actually for the last over 20 years really. And so we've had different generations from a single tube to like these more sophisticated plate readers. And so I've sort of followed the evolution of this type of equipment as we developed the technology for the use of our viruses as well. The lab has one main direction of work that we're planning forward in the next few years, and that is really to keep working on the topic of understanding transmission, mainly by mosquitoes, but also ticks to some extent. And we're trying to understand how these cells and how these vectors, so we were also working on the live insect, how they respond to a virus as being there, so a virus replicating, but also how the virus interacts with the cells in these vectors. and that is to get a deeper understanding of how they transmit viruses. Arboviruses are present worldwide, but I think what we've seen over the last 10, 15 years is that we've seen viruses emerge in places or parts of the world where they were not before. And a good example of that, for example, is Zika virus when it emerged in Brazil eventually. In the last few years, we've seen Zika, dengue, chikungunya also being transmitted in Europe, and although these are very small outbreaks, it shows that there's an increasing risk from arboviruses. And so there's many factors that feed into this, and this is something that is nonetheless, I think, putting these viruses in the spotlight again. It shows the relevance of this type of work and the impact it may have really worldwide.